thank you everyone here and online for sticking with us. I'm really fortunate that a few people that are more technically literate with PCs were here in the room with me, so we've got a workaround here. Okay. So, yeah, no, it's just going to be weird. Okay. So I wanted to echo the acknowledgement that was done at the very beginning. To just say that my lab is very grateful for the privilege of working, and myself and some of my students used to live as well on the traditional ancestral and unceded territory of the Musqueam people up at UBC main campus. So, you know, I've been very privileged during my career. Sorry for those of you in the room, you're going to hear this again. I've been very privileged to live on some really beautiful lands during my career. And so I thought I'd just start off by sharing a little bit about where I've been, and then we can talk about where the lab's going and what the lab's interests are. So I grew up in Ontario, and then, you know, I had done a lot in Southern Ontario. I really enjoyed life there. And then I decided it was time for a change, and graduate school was a great time to make that change for me. So I moved to the University of Glasgow to do a Wellcome Trust PhD fellowship, and I lived there for a few years. And it was a really wonderful experience living in Scotland. I did lots of beautiful hiking and experienced lots of really amazing food there. If you ever go and you're not vegetarian, haggis is a real, it's a very interesting treat. So from after I lived in Glasgow, I wanted to move back to Canada, and I decided to sort of move further west than I had been before. So I moved to Calgary to work with Dr. Savraj Graywal. And, you know, I did a lot more hiking there. It's a big part of my life being outdoors. And so I hope that all of you who have hobbies are able to indulge in them as well, because I think it's very important for your health as a scientist and also as a person. So after I lived in Calgary, I wasn't sure if I wanted to, you know, move on from being a postdoc and take a job as a professor or not. But I applied for one position, and I was fortunate enough to get it. And so I moved to Vancouver in 2015. So I've been here ever since, and I plan to stay. I really love being in Vancouver. And so, you know, the biggest thing that people in the room have already heard that I just want to emphasize to everyone is that at nowhere along the way did I definitely think that I would have ended up in this job. So I hope that all of you are able to remain open and you know, do what's right for you at each career stage, whether that involves exploring alternative careers, because I think there's so many wonderful things you can do with science degrees nowadays. So if you ever want to chat about that in more detail, please don't hesitate to drop me an email. And if I don't respond right away, just prompt me again. Okay. So we'll start off with a poll, and people in the room have actually answered this poll. So if you're online, hopefully you're able to go online to slido.com, so you can just put in this number here, or you can use the QR code if it does work on your screen. And crap. So I have to be able to go to Slido. That's the thing where this is going to fall apart. So if you put your poll answers in here, because I'm not on my own laptop, I will read them out as I can see them. So the focus of my work is biological sex is a variable when studying disease. And so I wanted to acknowledge that gender plays a really important role in health outcomes as well. It's not the focus of my research, but I do hope that all of you, with everything I say about biological sex today, will also, if your research, you know, considers gender or whatever, I think, you know, considering gender as a variable in biomedical research is just as important as considering biological sex. And the intersection between those two variables is also really an essential thing to study in the future. So, but for the purposes of today's talk, because it's much more my expertise, I'd love to hear from all of you about why you think it's important to consider biological sex as a variable when studying disease or even normal physiology. So we've got lots of really great answers here, and keep the answers coming if you're online. So we have sex can affect experimental outcomes due to differences in chromosome complement. So that's absolutely true. Sex chromosomes do definitely influence biology, circulating hormones, and all kinds of other things. Biological sex is important because there might be different dosage needs for medications. So I totally agree with that. Men and women and males and females do not always have the same treatment effects in response to different drugs. And even, one sex or the other can actually experience bad side effects of some treatments. So biological sex can affect response to treatments, can affect the results. So yes, exactly. So any results, whether that be RNA-seq, whether it be pharmacology, literally anything you do can be influenced by biological sex. There's sex and gender interactions, so I completely agree with that. It won't be the focus of today's talk, but I really agree with whoever put that. These are very essential things to study. You want to study biological sex and how it interacts with genes, hormones, and other pathways and systems of biology that relate to health and disease. So yeah, signaling pathways, different genes that put you at risk, put individuals at risk for developing different diseases. It's also important to know if biological sex doesn't associate with a particular disease. That's very true, because then you can have individuals that are male or female, and they can be treated in the same way if there is no effect of biological sex. The hormones associated with biological sex can impact the way the body functions, yes, and in order to support health equity for all people. And I think that's very true. And it's really important to remember, the individual here put for people of all sexes. And I think it's really important to remember that although today the animal work that we've done and the small amount of data in humans that we have as well, we've categorized that into male and female because that's primarily what our system works with. But I think it's important to recognize there is variation. There is a spectrum in terms of biological sex, in terms of the number of sex chromosomes an individual may possess. There's diversity in that in humans. And also there's diversity in terms of the amounts of sex hormones that individuals will have. So it's really important to remember that biological sex, like gender, is not binary. I will be treating it as such just because of the nature of the studies that we do. However, it is important to recognize that it genuinely is not. Okay, so I'll keep moving on from that initial slide. We'll do another poll later. So the focus of my research is metabolic disease. Now that's very, very broad, but I just want to introduce you to the idea that men and women differ in the incidence, progression, and treatment outcomes for metabolic diseases. So this includes things like diabetes, which is a dysregulation of glucose homeostasis in the body. Heart disease that's associated with diabetes. You can see here that women have a far higher risk. Women living with type 2 diabetes have a far higher risk of developing diabetic heart disease than men living with type 2 diabetes. Men are actually at a higher risk of developing type 2 diabetes, but in adulthood. That difference isn't so pronounced in children or post-menopause in women, but it is quite pronounced for both type 1 and type 2 diabetes, this bias towards men in adult individuals. And also when we look at cardiovascular disease, typically men have a higher risk of developing cardiovascular disease. It's about twice as high in men than in women. So again, I just want to acknowledge that, these differences that we see between men and women, there's two things that can contribute to that. Gender is one of them, because that can affect health seeking behaviors, the attitudes of healthcare providers, dietary choices, lifestyle preference, exercise choices, and things like that. But a lot of these trends that we see in people are actually seen as well in animals that don't have these social and cultural attributes of gender. So it really suggests that alongside gender, biological sex does play an important role in this sex-biased risk of developing different types of metabolic disease. However, despite the fact that these differences have been pretty widely reported now, there really are a lot of studies at this point. There's sex differences in the incidence, progression, and treatment responses to many metabolic diseases. The clinical guidelines for preventing and treating metabolic disease are really pretty much the same in both sexes. It's not, men and women don't get different prevention strategies or different treatment strategies for the most part. And the reason for this, one of the reasons for this, is that we really don't have the information that we need. We don't have the evidence base from the literature to really be able to actually start to develop these sex-informed prevention and treatment guidelines. So in my lab, and others have done this in different fields, but in my lab, we did a huge literature review in papers published in this journal called Diabetes. It's really the flagship journal of the American Diabetes Association. And we read all papers published in 1999, 2009, and 2019. And we scored these papers according to whether or not they included biological sex as a variable properly. And what we found when we scored these studies, where we found 0 is basically like not including both sexes at all, not even mentioning the word biological sex. A score of 1 would be, they just used males only, for example, but they didn't say why. A score of 2 would be, they said why, they would include just a single sex. A score of 3 is just a mix of males and females, so we have no idea what numbers of each. And then 4 to 6 were varying degrees of, including sex-specific analysis. So for example, a study could score a 4 if it had one piece of data, so one graph that showed males and females. So it's kind of a low bar. But what I hope you can appreciate is even as recently as 2019, fewer than 15% of studies actually even had one graph showing males and females separately. So really, there's a lot of room for improvement in terms of inclusion of biological sex as a variable in diabetes research. And if we had done the same with gender, I think, generally we would have seen the same thing and possibly even worse. And if we look at, in these single sex studies, if we look at what sex is sort of used more frequently in these studies on metabolic disease, and in particular focused on diabetes, we see that fewer than 12% of these single sex studies were done on females. So together, these two things really show you that not only is there a lack of consideration of biological sex as a variable in diabetes research generally, but we really particularly lack a lot of information about how female animals, female physiology, behave in response to a number of genetic and pharmacological treatments. So there's really a lot left to learn. And if you want to know more details, we've put this on bioRxiv now, this literature review. Okay, so people aren't doing enough of this, and this is the field of diabetes research. The same has been done in neuroscience, psychiatry, and some other fields as well. So all of you are going to be, working in very different fields. So I'd love you to go back to the Slido if you can, and to actually, put down, if you can, some of the barriers that you face in your own research, or, some things that have come up in your own research, or things that you've heard at meetings or conferences or whatever. So what do you think are the barriers to better inclusion? Because it's clear that we're not doing this good enough. So what are some barriers that you can think of? And then for everyone online, I'll read them afterwards. Okay, I can see people typing. This is good. And remember, all these answers are anonymous, so there's no information there. So I'm seeing some really interesting answers. So we have, it's hard to do statistical tests if there, a bunch of answers just popped up. Hard to do statistical tests if there are too many bins of different categories. So that can be true when you have, if you have like a rare population or something, it is more difficult to get enough participants to, keep subdividing everyone. So that's true. That can be a challenge, yeah. So just having to figuring out how many people or how many animals are needed to do a certain study, population sampling availability, lack of resources to support or recruit more diverse participants. So hormone cycles are different. That's a really interesting one. Limitations of funding opportunities. So yeah, lack of funds to do larger studies is a big barrier. Built-in and established structures and processes that are difficult to change. So I totally agree with that one. Actually, I've had conversations with folks who have done research. They found sex differences, and then some of their co-authors actually pushed back and said, oh, but we can't do that. That'll make everything too complicated. So we just won't include that analysis in there. And they, just went ahead anyway. So it's true, the sort of culture in some fields is to just not have to deal with biological sex as a variable. And then the gender versus sex debate. So when you're working with human data from humans, that can be difficult. So how do you assess each variable? I would argue that both should be included and both should be considered as full variables. But it is, when you're starting out, deciding how study design, that can be complicated. So I'll wait. I don't think there's any, there's no word. Oh, weaponization of data, lack of justification for funding. And we have less data and research on one sex compared to the other. So I think that's actually a really good point. That's a known barrier, the fact that nobody publishes work from both sexes. We have this lack of knowledge, and I showed you in the field of diabetes research, that lack of knowledge is in females. So now you can't explain some of your phenomenon that are different from what's published because you don't have a body of literature on female animals to compare that to, or to show that that's actually normal for females, even if it may not be normal for males. Inconsistent measurement of sex. Yeah, self-reporting genetic data, hormone levels. That's very true. And confusion from reviewers. So everybody can get pushback from reviewers saying, I don't know why, I've had this myself, I don't know why you're doing this. There's no reason why this should be different. And so that's a very difficult thing to actually go back to a journal with. So this is a really great discussion. I'm really glad people have had the time to think about what some of these barriers are. So there are a lot. And so a lot of people don't do this, but it's important. And so I hope over the next little while, I can start to show you how we've tried to make some inroads into this problem and suggest some opportunities that everyone can hopefully incorporate into their own work going forward. So the first thing I just want to sort of put out there is that, when we're used to, I'm a biomedical researcher, so, some of you might be like population scientists or epidemiologists or what have you, but many people that are doing biomedical research don't always, appreciate what you would see if there was a sex difference between the sexes. And I'll give you an example of that. So for example, sometimes, people have an animal model and they'll see that, the female animals will have a big delay in getting the phenotype compared with the male animals. And they'll tell me, well, but that's not a difference because the female animals eventually get the phenotype that, we're talking about. However, if, that bar would be completely different if they were looking at a different genotype and they would say, oh, this genotype, we can delay the onset of the disease in this genotype, and that means that genotype is protected. So they have kind of different ways of assessing their data depending on, whether it's a different genotype. Oh, look, we've delayed the disease onset. But in females, it's not a delay. It's the same thing because they do get the disease anyway. So it's the same phenotype. So what I would argue for everyone in this room, how I define a sex difference really is that it's any non-identical response to a genetic, pharmacological, or environmental, or otherwise manipulation or exposure or whatever. So it's any non-identical response. So if the timing of the response is different, if the onset or the symptoms are different, all of these things, to me, represent a sex difference. And so, for example, things that you might see in your data, if you have, I call them like class, so I've divided them into 4 classes. So, class 1, you might see one sex, for example, doesn't show any response to some diet, and there's a huge response of the other sex to that diet. So that's a very sex-specific phenotype. So one sex responds, the other sex doesn't. On the other hand, you might get a sex-biased response. So one sex responds a little bit, the other sex responds a lot. So they both kind of have the same direction of effect, but the magnitude is very different. Very rarely, you do see sometimes that one sex will go in one direction of effect and the other sex will go completely in the opposite direction. So completely opposite direction of effect. And then sometimes, this is something we've seen in my lab a lot actually, is that both sexes, we've seen this in a couple of projects anyway, both sexes have the same response, but when we unpick the molecular and genetic underpinnings of that response, it's actually completely different. So those are the scary ones, because your temptation would be to ignore what's actually going on here, because at the surface level, there's no difference. But when you unpack the really detailed mechanisms underlying the phenotype or response or whatever you're looking at, it's different. And so it really suggests that males and females really should be included at all stages of the study that you're doing, because you really can't predict where the differences are going to lie. The phenotype might be the same, but the underlying pathways or the underlying pharmacology or what have you might be different. So I hope that that will help frame people's thoughts on, if you do see something different, how do you kind of put your finger on what the difference actually is? Is there something sex specific going on? Is it sex biased? This is actually called cryptic effects. So are they sort of opposite? Or is there no difference on the surface, but perhaps there still is a difference if people were to do further, deeper investigations? And then just to show you, when we talk about how obvious sex differences are, it's very rare that there's this all or nothing effect. So sometimes people in biomedical science, they'll knock out a gene or you'll give a drug or what have you, and you expect to see a very large effect. And sometimes the magnitude of effect between males and females, the difference isn't huge. And I think it's something you have to really get your head around. When you look at data, they can look similar, but if you do the right statistics, you can actually unpack that there is actually a difference going on there. So I'm showing you some examples on the top, 2 of them from my own work, and then on the bottom from another paper. So for example, on the top left here, what I hope you can see is that females and males, in response to a drug, they decrease the levels of protein synthesis. And they actually, our stats show that they do this largely to the same degree and the same extent. However, females actually recover and rebound protein synthesis after this treatment, whereas males actually don't show that rebound. So again, if you were just to glance at the shape of these graphs, you'd be like, yeah, it looks kind of the same. But when you go and do the statistics, so in this case, it was a 2A ANOVA, you really can see that the quality of the response is different. Females are decreasing protein synthesis or whatever phenotype you might be looking at, and then they're rebounding, whereas males decrease initially, same thing as females, but they're not able to rebound. And so statistics helped us to pick that apart, what was actually going on here. Similarly, we're looking at static glucose-stimulated insulin secretion. So circulating glucose is really kept at within a very narrow range, and insulin levels are similarly very tightly controlled in circulation. So the variation that you sometimes see in the levels of the hormone between the sexes is not huge. But again, we can use our statistics to unpack the fact that females are showing a much larger response here. They're able to maintain glucose-stimulated insulin secretion. They have a larger magnitude of an increase, and they sort of return to baseline after this drug treatment we applied, whereas males overall did not show any kind of increase and in fact ended up lower in terms of their baseline insulin secretion compared with females. So again, if you just look at these graphs, they don't look that different. But when you use statistics, you can really unpack that there are different things going on here. Because when we use a 2A ANOVA, we can see that there's a sex by treatment or sex by time or what have you interaction. And this is a very obvious phenotype. So again, blood glucose is really kept within a very tight range. And we have, the only genotype that's affected are these males, which develop severe diabetes quite quickly. Whereas the rest of the genotypes, the control males and the females, both the controls and of this mutant genotype, the females are completely fine. They maintain their blood glucose levels completely fine, whereas the males develop this severe diabetes. So there's all kinds of different sex differences that you can see, but I just really want to introduce the idea that they're not just these stark everything or nothing kind of differences, but they are physiologically important. Okay, so I'm going to, I don't know when we started.
Keep going. You're 12:49, so we officially started about 12:25.
Oh, okay. I'm just trying to think, I want to respect everyone's time. So I'm a little bit unsure. Should we try, is it more important to stick to time? I was going to say, like, I can actually, I mean, it makes, we got into the idea here.
We have it recorded as well, so we'll be able to distribute that as well.
I'm just wondering, like, I'm wondering whether I should just go through like 1 or 2 slides of just a summary slide rather than actually going through, because it's kind of like starting now and doing 20 minutes, which is too much. So I think, okay, so I'm just going to skip massively ahead. So the interests that we have in the lab are metabolic disease. Sorry, this is very, I don't know what to do. Yeah, it's very unusual. I don't really know what to, normally when I'm running short on time, it's my own fault. And so I adjust. Okay, so we're very interested in metabolic disease in my lab, and in particular, we're very interested in diabetes, which is the loss of glucose homeostasis. And one of the main reasons that for that loss of glucose homeostasis is that the cells that produce the hormone insulin, which maintains blood glucose levels, they become dysfunctional and eventually they die. And so we know that males and male animals and men are at a higher risk of developing diabetes. And so in my lab, we really wanted to sort of unpack differences in these insulin producing beta cells that are responsible for maintaining blood glucose to see whether there's sex differences in that cell type. So we wanted to do, because there's a lack of high quality data out there generally, we wanted to generate a lot of high quality data, both in humans and in rodents, to try and understand a little bit better what the sex differences in this really critical cell type are. And so hopefully it'll give you some ideas about what, if you're doing RNA-seq or anything like that, it'll give you some ideas about, the things that the kinds of things that we saw and the kinds of things that other people might see. Actually, I'll just talk about the human stuff, and then I'm going to leave it and take questions. We have a lot of rodent data, but I'm just going to leave it there for the sake of time. Okay. So we're very fortunate to have access to some human data. So this is beta cells. So these insulin producing beta cells, there's single cell RNA-seq data from males and females, both living with type 2 diabetes and in non-diabetic individuals. And what we did was we compared, is this big enough for everybody? I can see people leaning forward. Is this better? Oops. Is that better? Okay. And so we wanted to compare gene expression between men and women in the beta cells, just to get an idea of what the sex differences in beta cell gene expression would be, both in non-diabetic individuals and in people living with type 2 diabetes. And so what I've done here is, because we found very profound differences between the sexes in beta cell gene expression, we actually separated male and female beta cells to try and understand how beta cells from females are affected, the gene expression response of females living with type 2 diabetes is affected, and how the gene expression response of males living with type 2 diabetes is different. And so, okay, the animations aren't working, so they've all just come up at the same time, unfortunately. Don't know if I can't go back. Okay, so because the animations aren't working, it feels a bit strange to do this. So when we look at the gene expression response, long story short, it's very different. So for types of genes associated with certain cellular processes that are, for example, upregulated in males, so cellular responses to stimuli, what you can see in females living with type 2 diabetes is that genes associated with this process are very highly downregulated. So that's an example of a gene expression response where the gene expression response is going in completely the opposite way. So in males, those genes are upregulated, in females, they're downregulated. There's other types of genes where, for example, that belong to certain categories where you can see that the magnitude of the upregulation, I'm just trying to find the right color because the animation's not working. So one of these ones. Okay, so for another category, you can see that even though RE1 alpha activates chaperones, it doesn't matter if you appreciate what this process is at all. But for this process, you can see here, it's like 15-fold upregulated in males living with type 2 diabetes, whereas in females living with type 2 diabetes, it's only upregulated by about 3-fold. So the magnitude, even though the direction of the effect is going the same way in males and females, there's a really big difference in how strongly those genes are upregulated in males and females. And then finally, what I hope you can appreciate is for all these things in orange, there's actually tons of genes that belong to categories that are only dysregulated in one sex or the other in individuals living with type 2 diabetes. So it really suggests that the gene expression response in beta cells in people living with type 2 diabetes is really not shared between the sexes. There's huge differences. And because we know that many of these genes are linked with beta cell function, it suggests that there could be some differences in beta cell function in individuals living with type 2 diabetes. So to test that, we're actually fortunate to get some data from the Human Pancreas Analysis Program. And what we were able to look at was the function of these cells. So the function is defined by their ability to make the hormone, make and secrete the hormone insulin that they're supposed to in response to glucose, which is the hormone that normally stimulates insulin secretion. And so what I hope you can appreciate here is that in males, so males that are non-diabetic in light green here, they're able to secrete lots of insulin when given glucose and other amino acids and things like that. However, in males living with type 2 diabetes, you can see that this ability to secrete insulin is very strongly impaired, very, very strongly impaired. And that really, is a classical, hallmark, the inability to stimulate insulin when high levels of glucose are present is really sort of this classic feature of beta cell dysfunction that we see in people living with type 2 diabetes. However, when we took the females here, you can see these are females that are non-diabetic, and they have, robust glucose-stimulated insulin secretion. But actually, in females living with type 2 diabetes, you can see that there's actually not a significant decrease in glucose-stimulated insulin secretion. And so, I'm going to finish here because it really suggests to us a lot of drugs that are used to treat people living with type 2 diabetes. Not all of them, but some of them are actually focused on stimulating beta cell insulin production. And if you think about, the consequences of overstimulating beta cells, that can actually be detrimental to their health. So if there's not actually an underlying problem in females with glucose-stimulated insulin secretion, which is what our data suggests, you could potentially be giving females living with type 2 diabetes a drug that might actually overwork their beta cells and potentially lead to more dysfunction and not actually help. Again, there's a lot of limitations to this data. We don't have information on medications. We don't know what insulin sensitivity is like in the people that we looked at. We didn't have any information on treatment intensity. So there's a lot of caveats here, and we need to do a lot more work to understand this. But I hope this little story really illustrates the fact that when you just, take all the beta cells, what had been done previously with this data is just to take all the donors and put it all together. And what we would have seen was that we would have missed a lot of the gene expression response because, if certain types of genes are only responsive in one sex or the other, you wouldn't see that in a large pooled sample at all. You would miss a whole bunch of genes that might be really important for beta cell function in disease. And then also, if we talk about whether these cells are functional or not, and whether, you're fixing a problem when you're giving someone medication to treat their type 2 diabetes, you might be trying to fix a problem that actually in females, our data suggests doesn't exist. So I hope this illustrates generally why including biological sex as a variable in all research is very, very important to inform treatments in the future and also prevention efforts. So I'll finish there. I'm going to skip right to the end just to go to the acknowledgements because I'm sharing this information, but all of the people in the lab that did this work are the real superstars. And so I'm here on their behalf, but the people in the lab that do this work are absolutely amazing. And in particular, George Brownrigg did virtually all of the studies that I did show you today and all the mouse work that I didn't show you. And we have some talented people working. We've been very fortunate to collaborate with some people to move our work. A lot of our work is in fruit flies, but we've actually moved to mice as well as an additional model. So we've been fortunate to collaborate with all these folks on these projects. And then we've been fortunate to be able to get funding from all of these institutions. So that's been really wonderful. And as I take questions, I'm just going to put this up here because these are steps that everyone can actually take in the future. Because I really think it is a community effort. It requires all of us to start doing these things, small and big, to try and understand a little bit better, what are the effects of biological sex on different diseases or in health, so that we can gain a better, more precise understanding of disease mechanisms. So I'll put this here. And I just want to highlight, when we did our study, we were really fortunate at the journal that they had a policy change right when we were doing, in the year that we were doing this study. So we went back and read a few more papers, and they actually introduced a policy that you had to say the sex or gender, if it was known, of the people, of human donors. That was the only policy change. But that significantly changed how people were behaving. And what we showed as well was that actually, whether people got funding from NIH, which has policies that you have to include both sexes, that doesn't make any difference to whether or not people include both sexes. But journal policy changes actually did make a difference. So I just want to leave that idea with you, because this is something we're going to try and carry on. And it just shows you that if you're a reviewer, you can actually ask for this. And that is another way we can make change as a community. So I'll be happy to take questions. And I think it's 101. So I think we're okay. Yeah, great. Thanks.
The human studies, the sex-specific analyses, how often are they matched for confounders like age and stuff like that, or do they control for those things?
I mean, some of this. So a lot of the studies just. What's the best way to put it? A lot of the studies just do a little bit with age. So a lot of the people living with type 2 diabetes tend to be in an older age bracket. So we did use a whole age range because we didn't have as many donors. So the age range spans, I think the youngest individual is in their 20s and the oldest individual is in their 70s. So it's quite a big age range. And so that's why we didn't have enough people to actually match by age. So that is a very important thing to recognize. So that's something that going forward we would have to do, but it just requires time to get more and more donors to actually be able to answer that question, because you have to get age-matched donors. But a lot of studies will, talk about age or separate ages, but then they won't separate by sex. So, yeah. Okay. Well, hopefully it was clear, and if not.
I have one question.
Yeah.
I was interested because I've read that there's quite a strong genetic component to diabetes, and I was just wondering if anybody's looked at sex differences in inheritance. So, if your father had diabetes, are you more likely as a male to get it versus the daughter? Just wondering about the interplay of those types of studies.
That's a really good question. So are people online able to hear?
Yeah.
Are they able to hear the question?
Mm-hmm.
Yeah, okay. So yeah, I mean, the question is to whether or not, there's a lot of people now working on whether, if you have, for example, a parent who is diabetic, how does that influence, if the gestational parent is diabetic, so there's a lot of people working on that now. It does look like the studies in mice, it does look like there might be some differences between male and female offspring if the gestational parent is diabetic. And certainly genetic components are important, but I would say that we don't have a full understanding of that yet, because oftentimes the sex of the sort of offspring is not separated, right? And so, there are studies that show that, there are some SNPs that are associated, or some genetic variation at some loci that are associated with an increased risk of diabetes in one sex and not the other. But a lot of those studies don't actually, again, just do that sort of analysis where they separate males and females out and ask whether this SNP influences diabetes risk in men. or in women or what have you. It's just sort of this SNP influences diabetes risk. And it's not sort of, so there's a couple of studies that have started to do that, and there are some differences. There's also some genes that are associated with rare forms of diabetes. And it's really interesting because there's a couple that I'm aware of. One is MOF-A, this gene, and actually in individuals that have mutations in MOF-A, I think it's the males that get diabetes, and the females get a different condition called insulinomatosis. So there you actually have the same mutation because it's sort of, the brother and the sister. So you have, the same mutation, but you get two different outcomes depending on whether the offspring is a male or a female. So, but again, we don't have the full information on that kind of stuff because there's not that many studies. And some of these diseases that cause early onset diabetes are sort of genetically strong genetic risk factors for diabetes. They're rare. So, it's not always separated by sex. So, but yeah, there's indications of these things, that there are sex differences, but we don't fully understand them yet, I guess is what I would say. Yes, it's a really great question. Yeah.
Well, thank you. Thank you again.
Thanks for having me.